This episode of the Deep Thinkers podcast is brought to you by Bright Island Outreach. Bright Island Outreach is a group of individuals, many of them dentists or dental professionals, that go down into the Dominican Republic multiple times a year. They are networked with local dental schools down there, and so they take dental students that are from the DR or going to school in the DR, and they take mobile dental units and go into these impoverished areas to provide pro bono work for the good people there who otherwise wouldn't have access to good dental care. They start with education and they teach them about oral hygiene. They also do fillings, extractions, root canals, and cosmetics. So they do a nice wide variety of procedures to take care of the people down there. Like I said, they go down there multiple times a year. So it's not like they're just showing up and providing a service one time and then disappearing into the wind. They take care of these people down there and they make sure that they get the repeated dental work that they need. 100% of all the donations given to Bright Island Outreach goes to the cause. So the people that are working for it don't make any money off this. They just get out of it what they put into it, and it always makes you feel good to serve people and help other people, particularly when those people can't help themselves. So if you're looking for a charity to donate to, this is a great cause. All your money that you give will go to the cause. And if you want to get involved a little further, say if you have an adventurous spirit and you want to go down to the Dominican Republic with this group, they also let you do that. Dr. Eric has been doing this since he was a child. His dad is a dentist, and so his family would often do these trips, so he's well versed in experiences like these. So check them out at brightislandoutreach.org. This episode of the podcast is also brought to you by Maracaba Brazilian Jiu Jitsu. Andre Maracaba has been doing Brazilian Jiu Jitsu for 24 years. He's from Brazil originally. He moved to Arizona 7 years ago and has been coaching at Power MMA until it recently shut its doors. Andre is an excellent coach and a great human. I highly recommend anybody who wants to get involved in Brazilian Jiu Jitsu to reach out to him. His last name is spelled Maracaba, and he's on Facebook. You can either reach out to him personally or like his Maracaba BJJ group. 2017 was a very good year for Andre's team. It was the first time he ever took the state championship in both the kids and adults. So he's who I train with, and he's my instructor. He's my friend, and he is an excellent coach and an excellent human. If you have kids, Andre is an excellent kids coach. He's also great with adults. If you're looking for a new gym, if you're experienced, it's great to come. If you're totally rookie, totally green, have never done jiu-jitsu before, that's fine too. Pretty much my case. I had had a little bit of experience with it, but not for 20 years, and definitely not enough that I could call on and have not enough to give me a head start in anything. But I'm loving it. Andre's a great coach. And this episode of this podcast is brought to you by Maracaba BJJ. One last item of housekeeping before I go. Actually, no.
Hello, Planet Earth. We are here today with Dr. John Wisco. Dr. John, hello. How are you today?
Good. Good to be here, Jesse. Thanks for
Very good. Thank you for coming on.
Yeah.
We had a couple technical difficulties. John and I did it here a moment ago, but we have sorted those out, and we are rolling and ready to go. John, why don't you tell us a little bit about yourself, including your credentials, what you're involved in, where you went to school, and things like that.
Okay. Where do I start? Let's see. I am currently at Brigham Young University. I'm an associate professor here. I'm also an adjunct associate professor at the University of Utah School of Medicine. I came from, I'll just go backwards in history. I came from the David Geffen School of Medicine at UCLA, where I was an associate professor. And then before that, I completed a postdoctoral fellowship in radiology at Massachusetts General Hospital at Harvard MIT. And then I completed my doctoral work at Boston University School of Medicine. That was way back in 2003. So, yeah.
So you've got a pretty stacked resume here.
Well, I guess. I mean, I've been fortunate to have great mentors over the years.
For me, it sounds very impressive. But then again, my half a semester at MCC didn't set the bar very high as far as getting impressed, by titles and things like that.
Don't discount yourself. I mean, you're doing a radio show and I'm not.
and I have dozens of listeners across the globe. If I'm being kind to myself, so
all 24 at least.
yeah. My mom listens regularly. So
okay, good. Okay, good.
Yeah,
hey,
good.
mom's the best customer. Let's see. So I completed my degree in anatomy and neurobiology. I was trained in neuroscience, but I'd say I'm really more of a clinical anatomist, a clinical anatomy radiologist, if you will, and medical educator. So that's kind of how I frame myself these days.
Very cool.
Yeah.
Now, from what I understand, you are El Jefe at the lab you work at.
El Jefe?
Yes.
Okay.
Is this true?
Did you hear that from Alyssa by chance?
I don't know who Alyssa is.
Oh, Alyssa Dale.
Oh, yeah, I don't. I didn't hear that from her, no.
Oh, okay. That's pretty funny. Alyssa's a former student of mine, too. So.
Oh, right on. Very cool.
Yeah. So.
Yeah.
I guess. I do run a big lab, so it's a lot of fun. I have about 30, well, anywhere between 20 and 30 students in my lab a semester. And then I run the BYU anatomy labs, and I have 80 TAs that I train and manage.
Nice.
Teach 800 students a semester.
That's a lot, man. You're getting the good word out there.
Trying, I guess.
Right on. Now, when it comes to science, especially, the fields that you're in, your focus can be kind of like looking at a beach with grains of the sand. There's a million different little things to go into. What is your little niche specifically? What kind of questions are you trying to answer with science? What type of new ground are you trying to break in what fields?
Well, I have, because I'm too ADD to focus on any one thing, I actually have 3 major research umbrellas.
Okay.
First is my Alzheimer's disease research work, and I'm interested in discovering imaging, but when I say imaging, MRI biomarkers for Alzheimer's disease, which there's a lot of work going on in that field generally. Right now, you can't currently diagnose Alzheimer's disease. There's no specific test for it. There are tests to get you close to Alzheimer's type dementia, but that doesn't really separate that type of dementia from other dementias other than our best guess right now based on some anatomical and functional markers that we do know. So right now, the only way to be diagnosed with Alzheimer's disease is postmortem, which is obviously too late. So, so my work is to correlate postmortem histological markers with imaging biomarkers that we can translate to patients. And so I have a big project there, and I just had a PhD student graduate and present his work, which is really groundbreaking. So that's exciting.
Very cool.
And I've got some reviews coming out soon. So that's neat. So there's that.
When those reviews come out, you'll have to send me to them so I can post them on my social media sites as a follow-up to this podcast, if you'd like.
Yeah, sure. That'd be fine.
Great.
The other thing I work on is musculoskeletal nerve and muscle fiber mapping. So I'm really interested in the larynx as far as how it works, its innervation, its mucosal, which is the mucosa in the throat, its mucosal response to hydration, dehydration, muscular involvement with how that works in terms of glottic stenosis, all those things about basically how the larynx produces vocalization, which is more
Okay.
complex than you think. We actually don't know very much about it. It's kind of surprising. We all know the anatomy, but we don't know how it works. You know, it's kind of
Well, that's why we have scientists,
right.
observing and taking notes so that we can get there.
Slaving away, I know. You know, the larynx community is actually quite broad. I have a colleague.
Wait. There's a larynx community.
Oh, yes. Yes.
I'm so glad I had this podcast today just so I can learn about the larynx community.
Well, I mean, the brain community is huge, but the larynx community is actually rather small. But, well, small relatively speaking, but it's large in terms of a lot of people interested in this problem.
No, I'm just being silly. From a layman, it's just like, wow, okay, I never heard of kind of put like that before. The larynx community. Okay.
I know. You know, you hear of all these weird scientific communities, and so
but that's why you're here, though, is to bring awareness to that. We know that these communities exist.
Right. And because of that community, one of my favorite things to do is I got the chance to dissect a whale larynx.
Wow.
A humpback whale. It was pretty big, and it was So yeah, my colleague Joy Reidenberg at Mount Sinai School of Medicine was the one who's the premier world expert on whale larynx biology, and so we're good friends. So,
that's cool, and that's how whales make their
sounds
like that can go for miles and miles into the water.
Yeah, it's pretty cool.
That is pretty cool.
So there's that. I'm also interested in orthopedic injury. So I have teams of students working on major synovial joints, the shoulder, the elbow, the knee, the hip. And our interest is mapping muscles and nerves and correlating as well as we can to MRI to be able to help clinicians understand what's going on with those joints so that they help them with their surgical procedures. And then finally, my interest in medical education is quite large. I teach a lot of anatomy students, both at the undergraduate, anatomy and physiology actually, both at the undergraduate and medical and dental school levels. And so I'm really interested in how students learn and how I can train teachers, peer teachers, to help their peers learn. So one of the big things that I do is a program called Anatomy Academy. We train high school and college students to teach anatomy, physiology, and nutrition to elementary school children.
Yeah, I was watching some of your videos for that online.
Oh, yeah. I'm glad. I hope you enjoyed them. And
yeah, they were cool.
Yeah, it's a lot of fun. Most of our sites are here in Utah for obvious reasons because I'm here. We actually started in Los Angeles, and the program has actually moved nationally through colleagues who are interested in starting service learning programs at their own schools. So
nice.
A bunch of different states. And so it's a lot of fun. It's a great program. One of the things I'm most proud of is that the program is really taking off and helping these elementary school students prepare for their standardized exams, and they do that through engaged learning. And one of the demographics, two of the demographics I really like are underserved demographic and also our special needs demographic. And
nice.
They have both. Everyone's really embraced the program, but those two in particular have benefited quite a bit.
Very cool. Very cool. Yeah, I know I definitely benefited from some extra attention from teachers in the special needs area when I grew up. I wouldn't have graduated high school without resource and without, teachers tutoring me during the summertime. So that's some very, very important work you're doing there.
Thanks.
All right.
Excuse me.
Well, I didn't know that you did work with muscles and joints, really. So let me ask you about this, and you may say, well, I don't really know much about that, or you may know tons about it. Either way is cool, but
sure.
Stem cell research and stem cell injections into shoulders and things like that. Mm-hmm. I've heard there's some groundbreaking progress that's been made as far as repairing joints without having to have surgery. Do you know anything about that, or is that kind of a different field? Or
that's a totally different field from what I work in.
Yeah.
I mean, I can tell you some generalities. Well, stem cell research is a hot field. The difficult right now it's a lot of promise and not a lot of delivery. A lot of that is because we really don't know a whole lot about how stem cells work. Excuse me. We know the potential, And we know that if we can turn on certain genes, we can get these stem cells to the word is differentiate from being a multipotent type cell to something that's specific. So just injecting a stem cell into a joint is low probability of it working unless we can get that stem cell to respond exactly how we want it to respond. And that's the difficulty, right? You got to have the right cell that's ready to become the right tissue and to have its genes turned on to undergo the repair process. I mean, just that, the number of variables just included in that sentence is enormous.
Yeah.
So that's not to say it can't work. It's just that it's a hot field of research because it has an incredible amount of promise.
Right.
We just haven't unlocked all of that. And again, I'm not a major expert in that field. So, but that's the premise.
That's cool.
Yeah.
Right on. Yeah. Where was I going with that? Well, while we're talking about gene editing and things like that, have you heard much about CRISPR?
Yeah, so I have some colleagues that work with CRISPR. I don't use CRISPR.
Let's talk about this. I'm excited. I love the idea of CRISPR, and I love hearing about it from people who know more about it than I do. So,
well, I'm not one of them.
You probably know more about it than I do, though, John.
But I have lots of colleagues that use CRISPR. They love CRISPR. Anyway, I'm not sure what I could tell you, but go ahead, shoot.
How long before I can get eagle talons on my hands? No, I'm joking. That would actually probably would work against me after a while. I wouldn't be able to type that well with them. But
right, that'd be very difficult.
what are first of all, for people who don't understand what CRISPR is or if they've never heard of it, can you offer an explanation that doesn't sound quite so bro-y? I mean, my explanation is going to sound real bro-y, lots of dudes thrown out there, lots of "it's totally rads" thrown out there in my definition. Can you offer a definition, an explanation of it that's a little bit more scientific than mine?
Probably not, only because I'm not a molecular biologist, but I can tell you the general idea.
Yeah, let's do that.
So the idea is that you have a virus that is injected into, a virus is used as a vector to inject some DNA into a cell. And you're trying to get that DNA to be activated by the host cell to, I don't know if changing the genetics is the right word, as much as trying to get certain proteins to be transcribed and then translated and then working in that particular cell. So it's a newer technique of what's been known as gene therapy, if you will. Trying to get new DNA into the cell. But basically what happens is you add in the DNA and then it gets incorporated into the cell's normal transcription machinery. And transcription is the conversion of DNA into RNA, and then from RNA into a protein is translation. So
okay.
So the bottom line is you're trying to get new proteins into a cell. And you do that through a virus. How that exactly works, I'm not exactly sure because that's not my field of expertise.
Yeah, I have no idea how it works either. Oh, I've heard that it can be described, and this is probably way oversimplifying it, but it's almost like, say you've got the genome open, okay, and in this area you have the whole genome, but this specific little spot here, you have genetics that could lead to heart disease, and as an adult you have genetics that could lead to all manner of problems, just bad genetics based on the genetic roll of the dice. Mm-hmm. And from what I understand is they can metaphorically copy and paste good genes from someone who has a clear genetic path of heart, a good heart history, or a genome that's not predisposed to these medical conditions, and they can essentially copy and paste it from one person to the next and have some of those potential risks be cleared up because of.
Bobby Flay here. Oops, sorry. Yep.
It's all good. Does that sound accurate too, or am I a little off there? Or?
No, that's accurate. That's the idea. So we know the sequence of the human genome.
Right.
Sorry, Bobby Flay popped up there, by the way.
No, it's fine. I like Bobby Flay.
So we know the sequence of the human genome. So we also know where. So every gene is framed by certain base pair sequences, and so we know those sequences. So we can add in. We can add in pieces of DNA that can insert themselves into those regions. So that's exactly what you're talking about. And since we know the map and we know where to go, we can insert things. Of course, the issue is we're really only good at doing one at a time, one piece of DNA sequence at a time. And most of the diseases or genetic anomalies, I mean, there are some point mutations. Most of them are multifactorial, multi-genetic, if you will, in terms of there's many sites that when transcribed add up to RNA, and then when translate add up to a protein. And we simply don't know if one change in a little sequence of DNA will make that big of a difference. It may or may not.
Gotcha.
So it's because there's so many different ways in which cells transcribe and translate into mRNA and proteins. We're not sure if the promise is there. Certainly could be, right?
Right.
For those diseases which we know are point mutations, that's a no-brainer. That's easy. So
gotcha.
A point mutation is one base pair or one amino acid change.
Gotcha. So we're still very much in the earliest stages of really just figuring out what can be done with CRISPR even.
Oh yeah, but it's probably one of the ones that's moving the fastest in terms of its promise, because it's such an easy system. You use a virus, in its machinery, you deliver the DNA, and the cell takes over. It figures out where to put these things.
It sounds so cool.
Yeah, it's pretty neat.
So now I've also heard it described something like this, like in the future, hopefully they get it to the point where, say you and I both having brown eyes, if we wanted blue eyes, we would essentially get the CRISPR shot in the arm, and slowly over time our eyes would.
I think you're now talking about Planet of the Apes kind of stuff.
Is that it?
Yeah. so yeah, that is more science fiction than it is science.
Dang it, man. That's how I've been explaining CRISPR to people. Now I got to go find all these people I explained it to and tell them I was wrong.
Well, all you have to do is actually get some colored pair of contacts and then tell
I
them.
want my kids to have blue eyes. Come on, John. Brown eyes are so boring. Let's be honest.
But brown eyes are dominant.
That's true. That's true. But blue eyes are pretty.
Well, okay.
No, right on, man. So that's interesting. That could CRISPR be the future of not only treating genetic diseases and genetic disorders, but any type of disease in general?
Well, we've been saying things like that about every new technology that comes along as the years have come by, and here we are. I mean, we haven't figured out how to fix every disease yet, and a lot of the reason is that even though newer technologies like CRISPR get us closer and closer and closer, we simply don't understand everything about how a cell's machinery works. So the importance of basic science research is paramount to helping those technologies actually do their jobs. Typically a new technology that comes about, its utility really isn't realized for at least 10 or 20 years down the road, simply because we don't understand how it works, right? I mean, most of the antibiotics or medicines or therapies that we've been using for years, we really don't know how they work. And we're only discovering things, the basic mechanisms of things that were discovered, decades ago. So the answer to your question, is it the future? Yeah, it's the future. How soon is that future going to be? We don't know.
This is why we need more people in science. So we can keep studying down this.
And if I could advocate for people to participate in research studies, particularly those who have disorders or diseases, finding subjects that are clearly one disease or disorder versus another is very difficult to find. So most of our studies work with animals because we can control a lot of the variables. But
Right.
once you make the jump from even a rat to a human, that's a huge species jump. Even though,
Yeah.
so for instance, we use rats to study learning and memory because they're the best model for learning and memory. But to switch that to what a human can do or what we can fix in a human, super complicated. So the more good human subjects we have, the better the research progresses.
So you're saying if someone has a specific condition, they're diagnosed, they're being treated, they know they have this condition,
Yeah.
then you would encourage that person to consider becoming more a part of medical research?
Absolutely, absolutely, because it's just so hard to find the right subjects that we can do controlled experiments with. Just even, and I'm just talking about non-invasive things, just even,
Right.
Imaging or simple things like blood tests and anything we can do to be able to further the research in humans would be a huge step forward because there's only so far we can get with animals.
And don't most medical research places compensate people for their time when they come in?
Yeah, it depends on the study, but some studies you get lunch paid for. Some studies you can get thousands of dollars depending on the study.
But no, that's actually a unique thing I hadn't considered before. That actually we should, as a species, be, as a civilization, be very interested in medical research in general and participating in it. That's an interesting perspective there that I hadn't thought of before.
Yeah. So if you're not going to get a whole lot of people being volunteers for CRISPR technologies simply because
Right.
it's complex, and so that's going to be reserved to humans for right now. But let's say
for animals.
sorry, for animals, yeah. But let's say that we do have some therapies that have worked in animals, but we don't have the subjects to try them on for a clinical trial. We don't have enough. We don't have enough subjects to do a statistical analysis. That's the tough part. And the translation from the animal to the human often has to wait for years just to get the appropriate number of subjects. So, I'm not saying that we should all of a sudden sign up to be part of mad science experiments, but
Right.
to go to check out your local research institution and see how you can get involved, particularly if you have something in your family that's genetic or if you have a disorder or a disease. No matter how common or how rare it is, we have a serious lack of human subjects.
Okay. So as far as being able to find your local research center, what if my listeners are going to go to Google right now, what are some of the keywords they should type in that will lead them to the right direction?
Well, all they have to do is type in the name of the institution that is nearest to their hometown. And typically there's a research page somewhere on that website where it says, are you looking to participate in research projects?
So they can type in something like medical research institutions by state.
Yeah, or let's say, you're in Arizona, so if you typed in Mayo Clinic
Gotcha.
research opportunities, something would pop.
Okay. So that, for Arizona people, that would be a good place to start, would be Mayo Clinic.
Yeah, Mayo is cutting edge.
Very cool.
Yeah.
Right on. Yeah, CRISPR is one of those things that I've heard a lot about it, but most of what I hear is the same stuff over and over again. So anytime I get something new on CRISPR, I'm like, let's talk about it. One of the things that it looks like might be one of the concerns with it, maybe you can expound on this a little bit. And of course, we're going to go back to my ridiculous examples. But like, say I wanted to have lobster hands for some reason, and they can edit the genome into the code into my genome so that I start to grow lobster hands. Isn't one of the concerns that the gene sequencing that allows my hands to grow into lobsters, couldn't that potentially unlock some new type of genetic disease or disorder? that we would have to study and find a workaround for, or is that my imagination running wild again? Which happens a lot.
Well, well, it's definitely the latter, but the principle you're asking about is an important one, and that is if we start messing around with the genome of a particular individual, can it cause other problems? The answer is definitely yes. The potential is there. And again, it goes back to what I said earlier. We really don't know how all of these things work together or interplay. So I have colleagues here at BYU that have been studying for decades on how limbs are formed, right? And there are certain cell signals that have to happen at certain times, sequences, certain targets. So if we insert a gene somewhere where that is happening in a developing even chicken, right? How does that affect
relative to the T-Rex?
Well, right, yeah. I guess so. Right. So how does that affect the rest of the organism? We simply don't know, right? And that's a very good reason why you don't want to get a CRISPR injection to grow your lobster hands, because those claws may come out of your nose.
Which would be not what I'm looking for at all.
No, it's not.
Yeah.
So but it does make for good science fiction. It does make
eagle talons coming out of my ears. That would do me no good.
Right, exactly. We're more likely to become X-Men than we are to get this CRISPR thing right for genetic modification.
Exactly. Well, that brings up the question: is what will once CRISPR 100 years into the future, once we've developed CRISPR and once we know everything about CRISPR, what will a Homo sapien look like, or what will a human look like? Will we have Homo sapiens, then other branches? Will we have Homo sapiens that can have night vision and infrared vision just based on their eyes and things like that? Will there be multiple races of humans based on CRISPR and everything that CRISPR's made available to us? So
yeah, again, that now you're diving into ethics instead of science.
Right.
Yeah.
Right. And of course, when we're talking about my imagination, especially when I'm talking with someone who knows a little bit about what they're talking about, you with CRISPR, lots of things, but that much. So stop me at any time that this is a mental avalanche. But so 400 years from now, when we've settled Europa, we've colonized Titan, how are we going to use CRISPR to adapt human beings to these new environments where gravity's different, the environment's different? Can you answer these questions for me, John?
No.
Okay.
I can tell you this: we'll have something better than CRISPR by that time.
Okay, cool. So I'll have a whole new field of research to geek out on. Yeah. This is good.
Yeah. Sorry, I can't answer that question.
I'm a little disappointed, but that's okay. I wasn't expecting an answer there. I just thought I'd throw it by you real quick, see if I can catch you off guard.
Okay, all right.
So now you do a lot of traveling for work and things like that. What are some of the other things, other than working with students and educating and supervising in labs, what are some of the things that you're involved in? I know you're a very busy man, and it seems like you're on the road, either traveling with your wife, who is my cousin. Full disclosure, John is my cousin-in-law. He's married to my first cousin. And so
it just killed my credibility right there.
Oh, no. I had Lincoln on a couple weeks ago. We were singing into the microphone. It was great.
That's too funny.
So, Lincoln sounded great. I sounded the way I sound, which is, whatever.
Okay.
I'm not to overshadow Lincoln too much. So, but you're traveling a lot. Why don't we jump on that a little bit and talk about some of the things that you do when you're not in the lab or the classroom?
Sure. So I would say 99% of my traveling involves spreading the word, if you will. So giving talks about any of the three areas of research that I discussed, doing trainings for other people, particularly in the medical education field, and then that's pretty. So most of the travel I do is a workation, if you will.
Yeah.
I like to hang out at places where I go to, and most of my students have become good friends because we work together quite a bit, and
very cool.
it's unavoidable. And so,
Yeah.
and they're really good colleagues. My students are the best and the brightest. And, I think any mentor would say the same about their students. So, yeah, most of it is conference work. I probably travel to 4 or 5 conferences a year, which spans from anywhere between April to November. This year, I took a group of students and colleagues on a special trip, which was not to a conference, but we studied art as far as how it relates to anatomy. So, and we went to Italy to study anatomy in the art form. And I took students who were artists, students who were anatomists, colleagues who were professional artists, and we had a lot of fun doing that project and studying art in Italy. I mean, what a great way to study Italy. It was fantastic.
Yeah, that sounds amazing.
It was awesome. It was 2 weeks of just, I mean, fun academia, if you will. It was,
for sure.
it was fantastic. But we did a lot of walking. Well, we walked 108 miles in 2 weeks.
Wow. Well, that in Italy with all that great pasta, the walking is probably necessary. So
yes, and the gelato, lots of gelato. Yeah, one of the highlights in Italy was really studying how the ancient artists really understood anatomy. One of my favorite places was the Borghese Museum in Rome. That was just to study the Bernini sculptures was a real highlight of mine. I have zero, and I mean zero, sculpture education. I didn't study art history at all, nothing, zero. And I was just blown away. It was one of the most amazing things I've seen. So that was cool. That was really neat. But most of my trips are, I regularly attend the International Association of Medical Science Educators meeting, the American Association of Anatomists, the American Association of Clinical Anatomists, Society for Neuroscience, Human Anatomy and Physiology Society, and every once in a while I'll go to something more specialized in any one of those research fields. So, I go to the trips not only to present my work, but to bring my students to present their work. Because it's very, very valuable for them as they are trying to get experience in research in the clinical fields, and they're applying for professional schools. So it's good experience for them to understand what this kind of life can entail.
Very cool.
Yeah. So I love to do it. It's a lot of fun.
Yeah. Well, every time I see you, it seems like you're talking about some new experiment or some new project or conference that you've been to. Sounds like you have a lot of fun with your job, and you're in a very important field.
Yeah, thanks. Yeah, I wouldn't do it if I wasn't having fun.
Well, let's talk a second about research, scientific research and medical research, and specifically about funding. I mean, you're with a university, so I imagine your funding is pretty much taken care of by the university, but how difficult is it for scientists to get research and funding for the majority of their work? From what I understand, it's very difficult, isn't it?
It's extremely difficult. So a typical professor is in what's called a soft money position,
Mm-hmm.
or a combination with a hard money position or a fully hard money position. So I am really fortunate that I'm in a hard money position. What that means is that the university pays my entire salary, and any research that I want to do, they've given me some little funds here and there, but I need to apply for grants. Typically NSF or NIH is the place where I typically go. But NSF is another option, and that's National Science Foundation or the National Institutes of Health. And to get funding from the federal government is super hard. So there are several institutes.
Think about it. If the government should be doing anything, it should be funding science.
Oh yeah, I mean, there's no question.
If you're concerned about the future, next generations doing things better, better energy, better agriculture, everything goes back to science.
Oh yeah,
a lot of people realize.
Right. There are two entities that fund science. The government is the major entity. The other entity would be the private sector. So, and I have received some private funding, which is nice. It's a different animal, but it's a same idea. You're trying to fund work that will ultimately help companies, and for them to be able to develop and innovate so that they can take things to market. There's a major gap between basic science and clinical science research and what actually gets translated into the market. And that gap is very similar to what we talked about earlier. It's about a 10-year gap. So what I discover now won't necessarily make it into being therapeutic for another 10 years. And the reason why is it has to go through all of these processes of vetting, clinical trials, et cetera, et cetera. The value that companies have in this process is shortening that gap. And we call that translational medicine. So in either case, you still have to have a funding source. So whether the company fully funds that research, which has its own problems, right? Because now a company is going to have some conflicting interests in that kind of research, but it still gets funded and it still gets done. Right. Versus the government, which moves at a snail's pace in terms of moving the research to a translatable type product. And so there has to be an important balance, an important partnership between the scientists, the government, and the private sector. I don't think science can really progress without those three working together, but the model has really been mostly scientists and government, and then the companies sort of pick up where these discoveries have been published and run with it. So that's the current model, and it's moving more towards a triumvirate type model, but it's very slow because scientists are skeptical of companies, the government is skeptical of companies are skeptical of the government, and you're trying to get three entities that don't really like working together to work together.
So, they don't really trust each other, but they know right. if they can work together, they can move a little quicker.
That's true. Yeah, and so funding is very, very tight at the National Institute of Aging. At one point, the funding, the people you had to be in the top 1% of proposals in order to get funded. That's ridiculously hard.
Yeah, that's 99% of stuff that gets applied for funding. Why would you even apply?
Well, and that's just it. I mean, just recently there was news they were going to increase the amount of funding in the NIA in order to really combat Alzheimer's disease. I think the goal is the year 2030 to completely eradicate it. We're not going to make it.
I was right now. A bit close.
Right, because, I mean, we've got funding issues, we've got politics, we've got scientific rigor. I mean, there's all sorts of issues. It's a good goal. It's a really good goal to meet. But we're not going to make it because of all the different factors that are happening.
Yeah.
Other institutes in the NIH have easier funding cutoff lines. They're 10%, 25%, and it changes depending on the number of people that apply. I mean, it's an economics issue, right? It's the number of people who apply, the amount of funds that are available, which is actually the major factor. If the government's not sending funds to be able to fund science, then scientists can't do their work, right? So now, what that has a social impact, because if you consider that at let's just say the funding level is at 10%. So out of every 10 scientists that apply for a grant, only one of them gets it. Well, what happens to the other 9?
Yeah.
So what happens is,
by the way, let's point out that the other 9's research isn't trivial. It's all important stuff.
It is all important stuff. And now I'm a big believer in the peer review process. I've been the beneficiary and also I've been axed because of the peer review process. So I've been on both sides, and I still advocate for that process because it's an important process that we as a scientific community know which projects are going to have the highest likelihood of success and which ones aren't. Right now,
peer reviewing, it's like taking a spelling test and never having it graded.
Yeah, sort of.
I mean, isn't it? I mean, isn't the peer review process essential for making sure that everything is working and correct?
Yes. Yeah, definitely. I mean, the peer review process is super important. If you had one entity that we're reviewing, there's going to be bias, and so now there's bias anyway. Don't get me wrong. There's certainly bias in the peer review process, but it's the most unbiased method, if you will. So I'm a big advocate for it. Again, I'm not always awarded, and I'm in that 1 to 10%, and that happens. But I'm willing to accept it because good science moves forward when colleagues understand what will work and what not. Now, that's the major advantage. The disadvantage is there is bias, there is politics, and some scientists don't want some certain people's work to move forward, and that is a reality. But you're going to find that in any field, not just science. So, hopefully minimizing that, I think the system works. The other disadvantage is that because we are looking for projects that work, then there's less of an incentive to fund projects that we know probably won't work, but the high-risk projects, but could yield important information. And so that's always been a critique of the peer review process. And
a lot of people don't realize that the scientific method being wrong is just as much a part of it as being right.
That's correct. But it's very difficult to get published when
you're wrong.
Right.
We know this doesn't work, so let's talk about it.
And there have been some attempts to have journals of non-significant science, if you will. Science that just, we tried it, here's the experiment, this didn't work. Because it'd be important for other scientists who are trying the same experiment to realize, well, okay, that didn't work, so I'm not going to do that. But those are things that don't get published, and that's a fault in science right now that we've got to be able to figure out, because that information, like you said, is very, very valuable.
Well, that would be a great journal if someone were to put that together and come up with it. The Journal of Failures.
The Journal of Failures, that's right.
A better name.
I mean, in baseball, we celebrate people who are successful 30% of the time. In science, we celebrate people who are successful 1% of the time.
Exactly. Yeah. A 30% batting average in science would be amazing.
Amazing. It would be amazing.
Yeah, Dr. Manhattan there from The Watchmen. His would be a little higher. His would be a little higher.
Right, a little higher.
But so that's interesting. Let's talk real quick a little bit about journals and things like that. Now, let me segue into that a little bit. One of my favorite podcasts that I listen to it every week, don't miss it, is The Skeptic's Guide to the Universe.
Okay.
With Stephen Novella, Bob Novella, Jay Novella, Cara Santamaria. There's Evan Bernstein. Don't want to forget Evan. But they're a group of skeptics that basically go through a lot of different articles and say, okay, this is what the cold, hard science means here. No, smelling farts does not cure cancer. This is what the science actually... Do you remember that article a couple of years back?
Yeah.
Yeah, they went through that and they kind of said, okay, well, here's what the science actually means. So let's talk a little bit about, basic scientists, clinical scientists, science communicators, and journals. I know it's kind of a broad topic right there, but...
Yeah, that's pretty big.
I think you'd be a good person to kind of break that down like a fraction. I guess a lot of people aren't aware that labs are encouraged to publish, and how often they're encouraged to publish. And so I think that's part of the reason why we get a lot of articles that are maybe not quite as in-depth as they should be, is because labs are encouraged to publish so often. But you know a little bit more about that than I do.
Yeah. So the journal process is a very important part of science. Essential. That it's both a mechanism and a metric. So it's the mechanism by which scientists publicize their experiments, and based on the peer review in the journals, colleagues who understand the science have determined, okay, yeah, this is great work, and this should be known by the entire scientific and social community. So journals provide that mechanism. It's a metric in the sense that our careers are measured by how often and in what quality journals we publish. So quantity and quality have an important balance. Those who are your world famous scientists, they're publishing in journals that have a metric called an impact factor. In other words, how often is an article cited in a particular journal. So Science and Nature have the highest impact factors. New England Journal of Medicine, these big JAMA, these big journals, because people read and they cite that work because they know that those quality journals have published quality stuff, and it's been vetted and we can trust it, right? But that doesn't mean that the paper is perfect. It doesn't mean that the experiment is perfect, nor does it mean that that particular paper is going to solve all of the world's problems through science, right? Because that's just not possible. It's the perfect science experiment doesn't exist. So now what many researchers will do is they will, we love our work, and it is devastating when somebody says our work is crap.
I can imagine it would be. Yeah.
But it happens, and I think when you sift through comments that you receive from peer reviewers and editors, and you sift through the stuff that actually, okay, this would improve my paper versus this is just a personal attack, a scientist has to make that judgment. And it still hurts either way. But again, with the peer review process, we were talking about grants earlier, but in this case, we're talking about papers. It helps because I've never had a peer-reviewed paper that ended up published that wasn't better than when I first submitted it. And that really gets to the heart of scientific accountability. And that's where the community is really important. And that accountability permeates all sorts of different fields, but in science, it's very, very important. So what a scientist will typically do is they will apply or submit their journal to the best journal that they think would be the best audience for their type of work. And if we get rejected, and we were asked to submit revisions and get published, fantastic. If we're just outright rejected, then we go to the next journal that's not quite as reputable. And we just keep going on down the line. So we have journals that are at the top, like, again, Science, Nature, New England Journal of Medicine, JAMA. And we have journals at the bottom. Typically these journals are just getting started. They'll publish my pet's scientific work.
Paltrow's Goop would probably be on the lower end of that journal spectrum. Well, yeah. Or maybe not on it at all.
Right. Right. And I've published in the different spectrum. I haven't published at the very top. I haven't published in the very bottom. But in the first or second standard deviation of reputable journals. And there's still value in that, because getting the word out there. I might have a paper that, okay, I got this experiment to work, but it wasn't quite there. It wasn't the best experiment. It wasn't the best results. But maybe someone else can build off of that. And so these lower tier journals are important for that. Now, this is my opinion, are important for that, because then people can build upon that and eventually get the science paper, right? We need some of that bad science out there so we can critique and go, this is awful, I can do better than this. Right? We need that. That's important.
Yeah. I think a big part of the scientific peer review method is just identifying, what is good, what is bad, and then getting the word out that this is good science or this is junk science or whatever the case may be.
Right. Yeah.
Very cool. Let's talk about science communicators for a little bit. I think I only was made aware that such a thing existed this year. I had no idea what a science communicator, it's an actual position and what it was.
Oh, okay. Yeah. How are you defining that? Because I've heard that phrase used different ways.
I'm typically someone who is looking at the data and then writing the article. So is what you're saying that a science communicator is typically the scientist doing the experiment, or is there a level removed?
No, typically people who write the papers. So the first author on any paper is the one who is in charge of that project, or at least did the majority of the scientific experiments, did the analysis, did the write-up, and wrote the actual paper. The last author on a paper is typically the mentor, unless the first author was the mentor, right? So and in the community, we know who is where. So if I'm the last author on a paper, my colleagues know that the first author is my student. If I'm the first author on a paper, then my colleagues know that the last author is probably one of my colleagues, one of my faculty colleagues. Okay. So it takes a team to be able to run the experiments, analyze all the data, and write it up. And the order in which someone's listed on a paper depends on their contribution to that work. And everybody who's listed on a paper had to have some sort of significant contribution to be able to list it on that. So how you're defining a science communicator is somebody who takes data from an experiment within that team, perhaps, and then writes it up. Well, that's typically the first author anyway. Okay. So, but then there are papers called meta-analyses, where an author will take a bunch of papers, will take all of their data, and will So basically a conglomeration of all the data that's out there that they could find and do another analysis. So by your definition, that would also be a science communicator. But I would still consider that someone who's doing secondary research, where they're doing a review and putting something new together. Actually, some would argue that that's primary research, but whereas a review would be secondary research. So anyway, then there are the science communicators who are gathering information for government entities, companies, other scientists. That's just a different definition of a science communicator.
Gotcha. Okay. I had a question right there, and then something popped up on the screen behind me and distracted me, and I lost it.
Oh, that's okay. Was it Bobby Flay?
No, it wasn't Bobby Flay. It's actually something I was working on earlier.
Okay.
What's that doing moving? Okay. No, but okay, that makes a little more sense. So a science communicator is just someone who's either close to the initial experiment or who is collecting data from multiple experiments to kind of put together a meta-analysis.
Yeah, I mean the term science communicator isn't used very often in the science because we're already doing all of that stuff. So
right. Well, I guess I get it from the skeptics a lot, because they'll go to an article and they'll talk about the science communicator who either wrote the article or
in that context, that's always the first author.
Okay, so those people are typically pretty close to the actual data then.
Yes, very close.
Okay.
Yeah.
Cool.
Otherwise, they wouldn't be listed as the first author.
Okay, that makes sense. What kind of? I listen to a lot of podcasts. We're on a podcast right now. Do you listen to podcasts, or you just find yourself speaking on them occasionally?
the one or two times that I've on a podcast. I don't listen to a whole lot of podcasts. Most of my time is spent in reading, reading articles. Podcasts are certainly interesting. I do like. I mean, if we include TED Talks as a podcast material, then I do like TED Talks. A lot of colleagues that I have run into in the past have given TED Talks, and they're really nice summaries. TED Talks to me are the social media version of a review paper, and sort of communicating, and going back to this term science communicator, sort of summarizing what many people have done in their work for a particular field, and here's where we're at so far. So I do like listening to those because it gives me ideas. It inspires me to pursue one direction or another, and so I do like them. So TED Talks, I guess you could include that.
Yeah, I think TED Talks.
A whole lot of other ones. Well, there are some ones that are produced by the universities. I have a deep interest in law also, so I was listening to Duke Law School podcasts a while ago, and I like stuff like that.
Right on. I have a friend of the show, Dr. Kevin Folta, who's a biotech guy. He has his own podcast called Talking Biotech.
Oh, cool.
If you ever want to check it out. He was just on last week, so you will be he was episode number 7. You'll be episode number 8.
Oh, cool.
He's a land grant scientist out at the University of Florida, and doing a lot with genetic engineering.
Oh, awesome! Yeah, cool field.
It was really interesting to talk to him. He's pretty heavily involved in his communities, like yourself. So.
He's the guy you want to talk about with CRISPR.
I think he does more with plants, though.
Oh, he's a plant guy?
Yeah,
well, even plants. You can do CRISPR in plants. So.
Can you? I guess, I guess you would be able to. Right? Yeah, cell. Okay. Wow. Okay. That just opened up a whole new can of worms for me to sit there and just kind of think about and be like, wow. So we can have roses that grow oranges. I cannot wait for this,
Well, keep in mind that most agriculture that we know today have been genetically engineered. Everything.
Dozens of times, yeah.
Yeah, I mean, right, and it's it's to improve, hopefully, theoretically, improve these things. Get them to grow faster. I mean, it's all about economics ultimately, right?
Right.
Christmas trees, corn, tomatoes. I mean, the cows we eat. I mean,
everything. Watermelon.
Right, watermelon.
Exactly.
Grapes.
Seedless grapes don't really pop up in the wild all that much.
Yeah, very difficult.
And bananas without seeds. I mean.
Yeah, I know, right? So
But
So
yeah, I'm always interested in food and where it comes from and how they're trying to improve food and what they're trying to do to make it better. The papaya, I think, is the rainbow papaya is the most recent masterpiece of genetic.
oh really? Oh, there you go. Yeah.
Well, the papaya was dying in Hawaii, and a local farmer modified it and brought in a gene from another organism that was resistant to this specific type of fungus or whatever was killing it.
Oh.
And not only is the papaya no longer dying in mass numbers, the plant, but it went from pink to more of a rainbowish color.
Oh, cool.
So they call it the rainbow papaya. But yeah, it's
Before
totally
long, it's going to be the kudzu of Hawaii.
yeah, exactly. But literally that saved papayas.
No, great.
Bananas that we eat right now are endangered. The dessert bananas that we all enjoy here because of disease. Same with citrus trees.
Yeah.
So people don't understand. People don't understand that the food that we eat now is not only has it been modified because of something in the past, whether it was trying to get more nutrients or more disease resistant or whatever it was changed in the past for a reason, and it's going to be changed in the future.
Right.
The world is constantly changing. So, as a species, we have to adapt with it.
Well, and that right there is the key is are we changing our environment faster than we can adapt to it? Right. So
Possibly, very possibly.
I mean,
Yeah.
let's just take wheat, right?
Yeah.
Wheat has been genetically modified in order to grow faster and more robust. And so, some people, this is all just, conjecture. Some people wonder if that has increased gluten intolerance. Right?
I heard Dr. Folta touch on that in his episode of the Joe Rogan Experience podcast, where he talks about how a lot of the from what I understand, gluten is the combination of two proteins that come together and don't break down as well when they come together. Now, I've heard what I think he mentioned is that it has to do with the size of the stalk in the wheat, that those amino acids that break down those proteins are actually built up in the stalk. And when we bred wheat down with a shorter stalk for convenience in harvesting, and I spent a couple weeks on a wheat farm, and we had amazing John Deere combines and everything like that to harvest that wheat. And I cannot imagine what it would be like to harvest wheat without one, particularly if the stalk is 7 feet tall.
Right.
But I believe that's what's caused, or it's speculated that that's what's caused a lot of the gluten intolerance with celiac disease.
Yeah. I mean, we don't know, but again, it goes back to this idea of if we're modifying organisms, what are the downstream effects that we simply don't understand?
Right. He does a lot of work with light too, where they shine lights on different foods and can kind of tell a food to do something post-harvest based on the light that they're
light activation.
Yeah.
That's where you put in a gene that responds to photons, and then it turns on something else. So
pretty interesting stuff. Pretty amazing stuff.
Yeah, that's cool stuff.
Now our time is. You've been gracious with your time, and we're starting to get a little bit close to the hour. One thing I did want to ask you, what kind of music do you like to listen to?
Well, I am a huge Beatles fan.
Ah, yes, my boy.
Yeah, I love the Beatles. I'll just name off my favorite bands. So Beatles are number one. U2's number two.
My first concert I ever went to was at ASU Stadium by myself. It was U2.
Oh, yeah.
Funny story. Your brother-in-law, Lincoln, was going on a date to that concert. I needed a ride. I was 13. None of my friends liked U2. Lincoln is the nicest guy in the world. Gave me a ride to the concert, gave me a ride back home, and then finished his date with the girl. Like she was a good sport, I knew who she was. She knew my brother, but
that's awesome.
It was pretty funny. I accompanied Lincoln and his date to that U2 concert back in 7th grade. But anyway, so U2 is a classic as well.
That's awesome. Yes, that's a great story. He's a great guy. Lincoln
is so cool.
Very, very talented man. He hasn't even really tapped his full potential, which is what's scary.
No. Yeah,
So
yeah. Lincoln is a time bomb of talent that's waiting to go off.
I know, right? Right. Um, so yeah, Beatles, U2, Billy Joel.
Love Billy Joel.
Yeah, huge Billy Joel fan. Then I'd say Sting probably
Okay.
next. Um, and then I like a lot of different bands. After that, I mean, I'll listen to those four all day, all the time.
Right.
After that, I kind of like I like Michael Jackson, Prince, um, Bon Jovi. Uh,
Yes.
you know, what's
I also
amazing.
went to his concert in high school.
That would have
Bought
been
a t-shirt, wore it to school the next day.
that would have been a great concert. I love Bon Jovi. Lately, so I like rock bands. I,
Uh-huh.
lately, though, I've really gotten into country, which I don't know
Oh,
where this has come from, but I know what it was. So I'm a huge Carrie Underwood fan, right?
okay.
She's the full package. Let's
Love Carrie Underwood.
yeah, I mean, you can't hold back about that. So I was just searching for Carrie Underwood songs and stuff on Apple Music, and I came across Keith Urban, and she did this really cool duet called "The Fighter" with Keith Urban,
Okay.
which is a fantastic song. So I said, "Oh, I should listen to some Keith Urban stuff." He's more of a, country rock kind of, sort of like the Brad Paisley type,
Yeah.
Garth Brooks,
Right.
yeah. And so I love his work. Keith Urban is awesome. and I never really gave him the time of day, I think, because I didn't really care for the whole Nicole Kidman thing. so,
Yeah.
so
He's a little too pretty for country music for me, man.
he's a little too pretty. Yeah, okay, all right.
Zach Brown. There we go. Zach Brown
Right.
looks like a
You
country-
like the burly guys, yeah.
Yeah, yeah.
So, but Keith Urban led me to Lady Antebellum somehow.
Okay.
And I love duet music anyway, right? So
Mhm.
anyone who does a duet, it's interesting to me because it typically tells some sort of story. And most of Lady Antebellum's music are stories, and I love it.
Right.
In fact, I've gotten so interested in it that some friends and we just recently went country line and swing dancing, and it
Nice.
is my favorite new thing to do.
There you go. There you go. Very cool.
It's been fun. So
Well, I've
yeah.
got some country music to recommend to you.
Okay.
Well, first, let me tell you a funny little story about Carrie Underwood. This did not happen in the first person. It's a third person story. I used to sell satellite door to door.
Mm-hmm.
And, you go knock on the person's door. They're interested in the product. You're saving them money. You're giving them more channels. They sign the agreement. Technician comes in right behind you and sets them up.
Yeah.
My buddy was this technician.
Mm-hmm.
And he was doing a direct TV install, and there's a direct TV channel that it just lands on when you program it, and it's usually a concert.
Oh, okay.
And someone's interviewing someone.
Yeah.
Well, this is the biggest TV on the market. This TV was massive, and right in the middle of the install, it pops onto this channel, and it's Carrie Underwood.
Oh,
Like
okay.
they're interviewing. And in his words, he said, she was the most beautiful thing I'd ever seen. And he didn't realize what he was doing, but before long, he found himself sitting on the couch in this customer's home and just watching the interview and the concert. And I don't know how long he was sitting there, but after a while, the homeowner shows up and goes, "Oh, do we like Carrie Underwood?" He stood up like, "Yeah, yeah." My bad, But yeah, it was pretty funny,
I have two good friends out here who met Carrie Underwood. I was so jealous.
Oh, right on.
Yeah. She's got a serious set of pipes.
Yeah, she's amazing. Shania Twain's got a serious set of pipes too. But my favorite country music singer of all time, Dolly.
Oh,
Love Dolly.
Well, here's the thing about Dolly. Not only is she a great singer, but she's a great writer.
Yeah.
She has written some of the most famous songs.
The Bodyguard theme song.
Yeah. I mean, that song is famous. And I don't think a lot of people know that about Dolly, about how great of a writer she is.
Oh, yeah, she's great. Yeah, she's an icon.
Yeah.
Some bands to check out. Look up a guy named Sturgill Simpson.
Okay.
Surgill, then Simpson. His album Sailor's Guide to Earth got Country Album of the Year last year.
Oh, yeah?
He is an outlaw country musician. He's got 3 albums now.
Okay.
And all 3 of them are great.
Okay. So in that vein, I really like Randy Travis.
Oh, Randy Travis is great.
He's, and of course, Johnny Cash. Those
Yeah, Johnny's great.
are classics. Oh, I forgot to mention Maroon 5. I'm a huge Maroon 5 fan.
Okay, cool.
I love their music.
Cool.
Yeah.
Cool. Yeah, Sturgill sounds a little bit more like Merle Haggard.
Okay.
Which, if you haven't listened to Merle, love Merle Haggard.
Well, a number of years ago, I had the opportunity to go to a conference in Nashville. And so the first place now we happened to be there at the same time as the Country Music Awards.
You did not meet Merle Haggard, did you?
Nuts. Yeah. So, I went to the Country Music Awards, or the Hall of Fame. Hall of Fame. And I remember reading up on Merle Haggard and some of the other folks there, and just was really, really impressed, for that genre of music that I really didn't have much background in.
Oh, yeah. I love the outlaw stuff. Shooter Jennings, Waylon's son, is amazing.
Oh, I didn't make that connection. Okay.
Yeah. Waylon. Well, his real name is Waylon Jennings Jr., but, his parents called him Shooter. Waylon Jennings is actually buried in Mesa Cemetery out there on
Oh, really? Oh, I didn't know that.
Off of Brown Road.
I didn't know he had Arizona roots.
Yeah, he married Jessie Coulter, and they spent the last 20 or 30 years of Waylon's life out here in Chandler. I'm pretty sure Jessie Coulter still lives in Chandler.
Okay.
So, but I would check out Sturgill, check out Shooter, Hank Williams III. Now, he's a little rowdy, so you probably won't want to listen to him with the kids in the car, just to help you.
Okay.
But he's got a couple different. He started playing with Danzig, which is heavy metal. He was, I think, their drummer or their bass player. From there, he went to a band called Arson Anthem, which is Phil Anselmo from Pantera's band. So he cut his teeth in the heavy metal industry, even though he is country music royalty. Yeah. From there, he signed a contract with a label and did a couple albums like his grandpa, which he hated. He's done a couple albums that sound a little bit more like his dad, but recently, since his contract's been up, he's done real folky, just outlaw music. Yeah. And also he has this genre called hellbilly, where he's mixed bluegrass with punk rock. And it's so unique, but it's really cool. It's a little startling, but once he gets into it, it's really cool to listen to.
That's cool.
That's called hellbilly. But, but it's great. See any other country music artists that I can throw your way?
Let me know.
If you like bluegrass, Old Crow Medicine Show is great. Yeah. The Devil Makes Three and Trampled by Turtles is all really good music as well.
Awesome.
So, yeah. All right, sir. We're getting pretty close to the end here. Do you have anything you want to plug, or any shout outs you want to throw out?
I don't think so. I think we covered a lot of cool stuff today.
Yeah, I'm excited to edit this and get this out on the airwaves so that dozens of people can hear it.
all 24.
All 24.
At least your mom. Yeah. My mom too now. Now you got
25, 26 if you count our moms.
Right, right, exactly. So no, thanks for doing this. I appreciate the opportunity. It's been good.
Thank you for coming on, John. I appreciate it.
Have me back anytime.
Yeah, absolutely. I'd like to have you on a couple times a year in the future. We'll probably have some more, some narrow fields of topic to talk about in the future.
Sure. And
if anybody has any questions for John, you can either tweet them to me on Twitter. I'm at Jesse Noble Smith, or if you have, I don't know if you field questions from the general public or not, but
yeah, I'd be happy to. I'm pretty sure my Twitter handle is at Jonathan Wisco, but it could be at JJ Wisco. Let me just check. At Jonathan Wisco.
Okay. All right, Jonathan, Dr. Wisco, thank you for your time. Thank you for coming on this podcast, and thank you, Planet Earth, for tuning in. Tune in again next time, and above all, be excellent to each other.